Bismillah ar-Rahman ar-Rahim. This is the last part in the revision of histological slides for the second year. This part involves the high power slides and the CNS slides. The high power slides are 9 in number. The first one is the seminiferous tubule. It is very easy to be diagnosed. Look for the tails of the sperms inside of this seminiferous tubule, and here we have small dark cells, spermatogonia, and these large cells, the primary spermatocytes together with the secondary and spermatids. The second slide is also very easy to be diagnosed. It is the Graafian follicle, and it is characterized by having this oocyte with the surrounding granulosa cells and the follicular cavity. And of course, we have here the zona pellucida, the nucleolus, and this part is a very characteristic point in the diagnosis of the mature Graafian follicle. Don't forget that Graafian is written G-R-A-A and not double F. Graafian follicle. This is a very easy slide. It is a slide for the retina. How to know the retina? The retina is formed of these layers of nuclei alternating with the acidophilic layers of the plexiform or reticular, and this is of course the rods and cones. So this is the outer nuclear and then plexiform, this is the inner nuclear and then plexiform, and then we have this ganglion cell layer. So these striations of nuclei is very characteristic for the retina. يبقى لما نلاقي الخطوط الزرقاء اللي هي عاملاها النيوكلياي متبادلة مع الريد areas, this is the retina. And here you will have 1 and 2, and then we have a very pale area for the ganglion cell nuclei. And these 3 are alternating with the red area. This is for rods and cones, this is the outer, and this is the inner reticular layers. Here is the cornea, and the cornea here is characterized by having the zone for the nuclei, one zone only for the nuclei, and then we have the parallel fibers of the substantia propria or the stroma of the cornea. These are red in color, and sometimes you can see in between the nuclei of the fibroblast, but it is not necessary. This is the organ of Corti, and it is characterized by having the cavities like this. We have this cavity, which is the scala vestibuli, as this is the vestibular membrane, and this is the cochlear duct, and this is the scala tympani. And the most important is the basilar membrane, and this tectorial membrane. This is the tectorial membrane like a hat, and underneath this hat we have the cells of the organ of Corti. So if you have this membrane here, this is the membrane basilar, and this is the vestibular, and here we have the scala vestibuli, here we have the scala tympani, and here we have the cochlear duct or the scala media. Here we have the tectorial membrane, which is supportive, and under this tectorial membrane we have the cells of the organ of Corti. This is the Malpighian renal corpuscle, characterized by the appearance of this tuft of capillaries that is surrounded by the space here, empty space, to receive the glomerular filtrate. And then we have the parietal layer of the Bowman's capsule lined by simply squamous. The most important thing is to look for the surrounding structures. The surrounding structures appears as tubules. يعني كل اللي حواليها عبارة عن أنابيب with lumen. So these are, here we have tubules, another tubule, with the lumen inside. This is very important. So it is surrounded by tubules. All of these are the tubules, the renal tubules surrounding this structure. So this is the Malpighian renal corpus. Don't forget, look at the surrounding structures. This is the islet of Langerhans, and actually some of the students make a mistake between the Malpighian renal corpuscle and the islet of Langerhans. How to differentiate? Here we have the islet of Langerhans. Here, this is the group of cells scattered. So you may diagnose this as the Malpighian renal corpuscle. And actually, sometimes you have also a very narrow white space surrounding this clump of cells, and you may say that it is the renal corpus. No, look at the surrounding tissue. Here at the surrounding tissue, we have the acini, and the acini do not show actually an evident lumen in the section like that of the Malpighian renal corpuscle surrounded by the renal tubules with the lumen. So, by looking to the surrounding tissue, you will notice that we have the acini, and actually the acini have a dark peripheral part or a basophilic peripheral part and an acidophilic central part. So here we have the picture like this, with no lumen inside. While in case of the renal corpuscle, we will have tubules surrounding. We will have these tubules surrounding the renal corpuscle with a lumen, and on the wall of the tubules you will see this row of cells. A very nice high power slide. It is the thyroid follicle. Very easy to be diagnosed. This is, of course, the lining cells, which are cubical or cuboidal, and inside we have the most important feature, which is the acidophilic colloid inside. Most probably we have this shrinkage, which is an artificial, leaving these striations inside. But don't depend on that, because sometimes, as here on the left side, we have the homogeneous colloid. So it may be showing striation, it may be homogeneous. This is not important. The most important, it is an acidophilic material inside this follicle, which is lined by one row of cubical cells with the rounded nuclei, or cuboidal cells with the rounded nuclei. This is the classic hepatic lobule, and it is actually taken from the liver of pig. Sometimes the view does not bring the boundaries. No problem. You will notice the central vein and the radiating cords of hepatocytes. So this is the classic hepatic lobule. Sometimes you will not see the boundaries because it does not appear in the field, but you should find the central vein. Now let's move to the 11 slides for the central nervous system. We have 11 slides. First of all, look for the central canal and the shape of the gray matter. Here we have a central canal and we have H-shaped gray matter. So first point is it is the spinal cord. Spinal cord. Then, which level? You will look for the shape of the horns, the outline of the section. Here you will notice that we have thin posterior, thick anterior, and very important, the divergence between the thin posterior horn. How to know the posterior from the anterior? The posterior reaches or touches or most near to the section, to the surface of the section, while the anterior has a great distance between it and the surface of the section. So this is the posterior and these are the anterior. Here the section more or less is oval, so this is the cervical level. So this is the cervical level of the spinal cord. This is a section showing a central canal, H-shaped gray matter. So first thing, it is the spinal cord. Then look for the following. Look for the shape of the horns. Here the horns are more or less of equal thickness. Here this is the posterior, of course, because it has a connection with the surface of the section, this connection, and this is the anterior. So the posterior together with the anterior almost similar to each other. And very important that here we have no divergence for the posterior horns. You see here that the posterior horns, the area in between, almost at right angle. This is a right angle and this is a right angle. So this is the thoracic. The four horns are equal and the posterior horns are almost parallel to each other, and the four horns are thin, very important. So this is the spinal cord, thoracic level. Also, the section is more or less rounded. We have few criteria here. We have the difference in the sizes of the horns. They are almost similar. The absence of divergence of the posterior horn. And number 3, we have the rounded structure, and it is actually a small section. In case of the lumbar, it is very easy. It looks like two half boots. This is the half boot, and this is another half boot. We have the four horns are thick, and first of all, as I have mentioned before, it is a spinal cord. Why? Because of the presence of a central canal and the H-shaped gray matter. So first word, spinal cord. Between two brackets, it is the lumbar. We have the four thick horns, and the posterior horns are thick and parallel. This slide actually here it does not show a central canal, but actually it is present in the section. We have a canal. So when we have a canal and the gray matter is not H-shaped, so it is closed medulla. How to differentiate between the closed medulla, motor decussation, and closed medulla sensory decussation? If you look here, you will notice that we have a bird here and his lower limbs here. This is like a beak for this bird. And the beak of the other bird, and the head and the wings, and his limbs. So if you have the picture of a butterfly or two birds, this is the motor decussation. Don't forget that the bird moves, so it is a motor decussation. Actually, what we have here, we have the direct pyramidal tracts after decussation has taken place, and here we have the gracile, cuneate, spinal trigeminal, all you know in the drawings of the CNS. So when you see a central canal but the H shape is not present, this is a closed medulla. Look for the shape of the gray matter. If it looks like a butterfly or two birds, it is the motor decussation. Here we have a central canal, as you see, and no H-shaped gray matter, so it is closed medulla. Type. Look at the gray matter. Here you cannot see the picture of a butterfly or two birds, but instead we have here anteriorly the full pyramids. And on each pyramid, you will have the medial accessory olivary nucleus, which is a gray matter, so it is dark compared to the pyramidal tract. Here they look as if you have a mustache. Here, as if we have a mustache. So this is the closed medulla sensory decussation. This is a section of the open medulla. The open medulla characterized by the absence of central canal. No central canal. Number one. Number two, when we look at the front part of the section, you will notice the appearance of the two pyramids, because here no motor decussation takes place and the whole pyramid is present with the accompanying medial accessory olivary nuclei looking like the shape of a mustache, exactly like that of the closed medulla sensory decussation, but no central canal. So it is similar to the closed medulla sensory with the absence of the central canal. Sometimes you will notice the presence of accompanying parts of the cerebellum and sometimes not. So it is not a permanent character. So the open medulla is characterized by absence of the central canal, but the pyramids are present in the same appearance like that of the closed medulla sensory decussation with the mustache formed by the medial accessory olivary nuclei. So we have here the pyramids completely present here with the surrounding medial accessory olivary nuclei. This section does not show any canal, so it is either the open medulla or the bones. Here we have the bones. Why? Because we have here the basis pontus. So if you have this plate, which is the basis pontus, and you have two peduncles like this, looking like a face of a cat, and sometimes we have parts of the cerebellum on the back of the slide and as well as on the sides. So this is the pons section in the pons. Very important that we have the basis pontus and the peduncles. In this section, you don't have a central canal, and we have another cavity, which is the cerebral aqueduct of Sylvius. Here you look for the cerebral aqueduct of Sylvius, so this is the midbrain. If you look for the anterior part of the section and you find the structure similar to the basis pontus, so this is very near to the pons, so this is the inferior level of the midbrain. More than that, you will notice that the sides of the gray matter are rhomboid, having this number of sides 1, 2, 3, 4, 5, 6 or 5. So if it is rhomboid shaped gray matter and we have the basis pontus still appearing here, so this is the level of midbrain inferior colliculus. Here also we have a cavity which is not a central canal, it is a big, so it is the cerebral aqueduct of Sylvius. And then look for the gray matter. You will notice that it has smooth edges, looks like an inverted pear. And then for the basis pedunculi, you will notice that we have two separate parts and we don't have the basis pontus like that of the inferior colliculus, so this is midbrain superior colliculus. This is a section for the cerebrum, and as you see we have the gray matter on the surface, not in the center as in the spinal cord. And the white matter is in the center. Here we have the gyri are very wide to compare this section from the section of cerebellum. Most commonly, this section comes stained by hematoxylin and eosine, but it may come also in silver. This is a section which is very easy. It is a section for the cerebellum, and you can see here the three layers. We have the molecular layer, we have the Purkinje and granular, and then the white matter of the cerebellum. Here we have the gyri are very narrow and they are branching like the leaves of a tree. This is a section of the cerebellum and it is most commonly stained by hematoxin and EOC. Now in this picture we can summarize the 11 CNS slides. First you look at the slide. If you see a central canal, then this slide is belonging to the group of the spinal cord and closed medulla. If the H-shaped gray matter is present in front of you, then this is a spinal cord. If no H-shape, then this is a closed medulla. In case of the spinal cord, look at the posterior horns. If the posterior and anterior are thick and looking like a half boot, this is the lumbar, spinal cord lumbar. If the posterior and anterior almost similar, and look at the angle between the posterior, they are not diverging, and the section is small in size. If the section and rounded, if the section is more or less oval, and the posterior horns are thin compared to the anterior, and they are diverging, it is the spinal cord cervical. Then, if no H-shaped gray matter is present, then it is a closed medulla. Here I am making another picture to show to you that this section has a central canal, but it is actually compressed or elongated, so it may not be evident, but actually the shape of the butterfly or the two birds are very evident. This is the head, the wings, and the tail, and the limb, and the head, the wing, the tail, and the limb. So they are very characteristic to the closed medulla motor. العصفور بيطير يعني بيعمل activity, motor activity، يبقى العصافير دي تبقى الموتور دي cassation. Then we have here the closed medulla because central canal no H, but we have here two pyramids and marked by this mustache made of the medial accessory olivary nucleus. So this is the closed medulla sensory decussation. Then no central canal. And what is the meaning of central canal? It is a small canal. Here I have a big canal which is not in the center. Almost, يعني it is a big and deviated posteriorly. So this is a midbrain. No canals at all, no canals at all. Try to differentiate between the midbrain. This is the superior colliculus in which we have the inverted pier gray matter. But this is the inferior. We have a rhomboid-shaped gray matter, and here we have a basis pontis. We have the basis pontis, but here we have the two crus cerebri completely separated from each other. But here you can see this angle of the basis pontis on which the midbrain rests, and it is very evident in front of this, in addition to the rhomboid-shaped gray matter. But here it is inverted pier, and even the central canal or the cerebral aqueduct of Sylvius looks like an inverted pier also. So this is the midbrain superior and this is the midbrain inferior. Then we have two sections which do not have cerebral aqueduct and they are not showing sulci and gyri. So this may be the open medulla and the pons. The open medulla is kidney shaped and showing the pyramids with the mustache. The pons is showing the appearance of a face of a cat. These are the two peduncles, inferior cerebellar peduncles, and this is the basis pontis, very characteristic like that here. And sometimes we see a cerebellum here, and sometimes also we see a cerebellum here, so it is not a constant feature, but this is a kidney shaped with the mustache, and this is a cat shape with the plate. Then finally we have the gyri, sulci. This is a cerebral cortex and this is a cerebellum. This is cerebrum and this is a cerebellum. We have here wide gyri and limited number. Here we have many gyri and they are looking like the tree، حتى بيسموها شجرة الحياة. تمام؟ يبقى إذا هنا ده اللي هو هيبقى السيربرم وده هيبقى السيربيلم. Best wishes for all of you.